You did. All right, can we think we're ready to go ahead and get started? So today we have Dr. Julie Schram, who is here as a faculty candidate for the UA position. Julie comes to us from the University of Oregon, where she's a postdoctoral researcher working in the lab of Dr. Erin Galloway. She's also a co-PI on a Oregon Sea Grant project where they are investigating the impacts of ocean acidification on Dungeness crab populations. Julie got her master's and PhD here in Alabama at UAB, where she worked with James McClintock, looking at how various invertebrates from temperate and polar regions cope with climate change. So I think we're ready to go ahead and get started. So today we have Dr. Julie Schram, who is here as a faculty candidate for the UA position. Julie comes to us from the University of Oregon, where she's a postdoctoral researcher working in the lab of Dr. Erin Galloway. She's also a co-PI on a Oregon Sea Grant project where they are investigating the impacts of ocean acidification on Dungeness crab populations. Julie got her master's and PhD here in Alabama.
My research has focused on organisms and how they interact with other species and their environment, and how these interactions influence their physiology, and that they have a finite ability to metabolize, get food, perform defenses, and they need to make those energy allocations based on what they have available and then their interactions with other species and their environment. And so to quantify this, I have used a bunch of different metrics, which include behavior, looking at the changes in biodiversity, and using trophic biomarkers. So what this looks like is this. So this word cloud, I generated it using my published abstracts. So you can see that there are some things that fall out. I particularly have looked at how changes in pH and ocean acidification influence organisms, as well as temperature. And these are a couple of the model organisms that I have used in my studies. They include everything from crustaceans to mollusks, all the way up to some fish, also some pyrosome work. Today I'm going to focus on some of my crustacean work, and I'm going to try to give you an idea of the scope of my work. So that will include some of the work that I did as a PhD student, some of the stuff I've done as a postdoc, and I'm continuing to do. So all of the places that I've worked are interesting to me for various reasons, and it's mostly because the environment changes and or doesn't change. So along the western Antarctic Peninsula, this is an area that has been historically very stable with respect to the environment, but now it's starting to change. And so how that influences organisms that have in the past experienced a very stable environment is really interesting to me. And then in contrast, my work on the west coast, the organisms experience seasonal upwelling. And so with that upwelling, you get a lot of nutrient flux as well as changes in pH. So there's a lot of low pH exposure every season. So I'm setting this up as a contrast. So here, very stable, not a lot of change annually. Here, a lot of change, and then looking at how organisms respond to that when I do other manipulations. Okay, so all of my work has been done mostly in the context of climate change. So this slide may not be necessary, but I like to start everything off by defining my terms. So when I'm talking about climate change, I'm referring to the collective environmental impacts driven directly or indirectly by the CO2 in the atmosphere. And so in a marine environment, this looks like ocean warming and acidification. And this is because the anthropogenic carbon dioxide, or the amount of carbon dioxide released by people activities, has been increasing at an unprecedented rate since the Industrial Revolution. Around the Industrial Revolution, the atmospheric CO2 was around 278 parts per million. Now it's over 400. I think the last figure I saw was around 420 parts per million. And all of this carbon dioxide being released into the atmosphere, it's not just hanging out there. About 1/3 of that carbon dioxide that gets released is absorbed by ocean waters. And so that sets up a lot of dynamics for organisms. So let's start off with talking about what people think of most when they think about climate change, and that is warming. So On this figure, you can see there are some faint colors. So these colors represent the change in temperature over a decade. So this is from 1970 to 1979. The cooler blue colors represent a cooling over that time period, and the warmer red colors represent a warming over that time period. And you can see that there is a mix across the planet. There's some warming, there's some cooling, there's not really big patterns, but when you fast forward to the time period between 2000 and 2009, you can see that that has dramatically shifted, gone to more of a warming pattern, particularly at the poles. And the area that I study is right here on the western Antarctic Peninsula. And you may not have noticed, but the peninsula had a slight warming trend before in the past, but now it has been greatly accelerated. And so I mentioned that, on the western Antarctic Peninsula, in the area that I work, I am lucky that there is a long-term ecological research, or LTER group, that has been working there for the last 45 years. And so they have been quantifying how this warming is affecting the communities. And the communities that they have been quantifying are focused more on these charismatic megafauna, the penguins, and along the western Antarctic Peninsula, particularly in the northern regions, you have a lot of these ice-dependent Adelie penguins. The Adelies rely on krill, which also, for part of their life history, they rely on this sea ice as a food item. And then they've not done as much with the benthic communities. However, what they've been noticing and what they predict will continue to happen is this. The sea ice is disappearing along with that warming, which results in a shift in the penguin populations. These ice-dependent penguins can no longer survive, and the areas that they used to inhabit are being invaded by invasive penguins, the gentoos and chinstraps. You also will notice that those ice-dependent food items, the krill, are gone, and you have other invasive species. One of the things that you might notice, or may not have picked up on, is that there's an increase in macroalgal cover when you get less sea ice, and that's because there's more light available. And this is the area that I am interested in. Since this habitat is going to be expanding, I'm interested in how the organisms that associate with that type of habitat will respond to warming changes. So the reason that these critters or organisms are interesting to me is because polar organisms are thought to be stenothermal. Stenothermal just is a fancy word for saying that they are adapted to a very narrow range in temperatures. So on this figure, on the y-axis, they have the aerobic performance and productivity, so all of the metabolic activities that these organisms can perform. And then on the x-axis, it has temperature. And you can see with the polar stenotherms, they have a very narrow range in temperatures where they can perform any of these activities that are necessary for life, especially when you compare it to temperate and tropical organisms. So this sets up the stage for a lot of these organisms to be sensitive to the changes that are already happening along the western Antarctic Peninsula. And to illustrate that, here are a couple of shelled species. These two species have a very narrow temperature tolerance. They can survive between -2 degrees C and positive 3.5 degrees C. And I can tell you that right now in the summertime, I just came back from Antarctica a week ago, and the water temperatures are already at 3.5 degrees C on the northern Antarctic, or the central Antarctic Peninsula. So these critters are already briefly being exposed to these temperatures. So this is already setting up problems for some organisms, but not all of them have such a narrow range. So that's why it's interesting to study them. All right. The other thing with carbon dioxide is that it gets absorbed by the ocean waters. I mentioned that before. And so you guys have you all heard of ocean acidification? Yes. Okay. Just in case you haven't been thinking about it, the chemistry as much as I have put together a little primer on what ocean acidification is and the chemistry. So basically, it refers to the CO2-induced reduction in seawater pH. And the way this happens is you get carbon dioxide and interacts with the surface waters, and that will form a weak acid, which is carbonic acid, which will quickly dissociate into a free bicarbonate ion and a free hydrogen ion. And just as a quick reminder, this free hydrogen ion and the accumulation of these free hydrogen ions, this is what triggers a change in pH. So as you get more free hydrogen ions, you get an increase in acidity and a decrease in pH. However, As you can imagine, this has been happening for a long time. There's a lot of carbon dioxide being absorbed by the surface waters. The ocean has a natural buffering system to deal with this accumulation in hydrogen ions. And so the way that typically works out is you have these free hydrogen ions able to combine with free carbonate ions, and they form bicarbonate. So now you don't have that accumulation of that hydrogen ion. However, this buffering system has been going on for quite a while, and these carbonate ions are becoming more limited than they used to be. So the rate of acidification is increasing as you run out of the availability of these free carbonate ions. That's not to say that they're limiting, it's just they're getting in shorter supply. So people have looked at some of the effects of pH on Antarctic organisms, and right now it's a mixed bag with the responses. So I mentioned krill are really important. They have exhibited some negative responses to decreases in pH. Most of them are sublethal. Those include elevated ingestion rate and metabolic rates, and there is some difficulties with egg hatching size. With the macroalgae, for the most part, the big fleshy macroalgae are fairly resistant for the ones that we have looked at, but to date, this is the most well-studied species. They can still do their photosynthesis and they can grow both at warming and at lower pH. However, when you get to encrusting seaweeds, then you get some interactive effects with temperature and pH that can negatively impact some of those species. All right, so when you think about Antarctica, this is probably what you're thinking about, right? Think of one of these organisms, or maybe multiple, the penguins and the whales and the seals. So I want to introduce you to the Antarctica that I know and love, and that is this. So right here, along the western Antarctic Peninsula, it's dominated between 5 and 40 meters by these big seaweeds. So you get, you might notice I use seaweed, macrophyte, and macroalgae interchangeably, just to note I am referring to the same type of thing. Okay, that you get these big brown seaweeds, like this one here, Hemantophalus. It looks like big strings of lasagna noodles that kind of drape down the benthos, and under and around those seaweeds you get a lot of these bushy red seaweeds. There are also a lot of big filament or branchy browns, and I'm highlighting these branchy brown seaweeds. This one here is Desmarestia menziesii, because they have a lot of amphipods and crustaceans that associate. Would find over 300,000. Don't is because the vast majority of these big brown seaweeds, they are chemically defended to prevent these amphipods from feeding on them. However, these amphipods still hang out on them because they get a refuge from predation. They are protected from other things that also don't want to hang out with bad-tasting seaweeds. And in turn, the amphipods eat the epiphytes. So there's a lot of epiphytic diatoms that could develop on these seaweeds, but they don't because you've got these amphipods just mowing them down. And this is considered a community-wide mutualism because of these abundant amphipods and these seaweeds working together. I'm highlighting this because this is one of my key systems that I've worked with. All right, so you might wonder if of the benthos dominant peninsula, this is where Anvers Island is, and the vast majority of my work has happened at Palmer Station. This is Palmer Station. You can now see all of it. It can have an LTER, and I have benefited a lot from their data collection. So I'm going to be showing some of the data that I've aggregated from what they've made available. So this here is seawater temperature data. So on the y-axis, I have the temperature in degrees C, and then time in months, and each one of these lines represents a different year of data. So I'm highlighting this because I want to show that I've been talking about how stable this environment is, but it does vary over time. So seasonally, there are shifts in temperature, and even day to day, there are shifts in temperature. They're just not as big as what you might see here. Another thing that the LTER quantifies is the chlorophyll A concentrations in the water column, and they use that as a proxy for phytoplankton. So since I'd worked for the LTER after I was an undergraduate student, I knew that these data existed and I knew how they collected these data. So for 1 year, I paired bottle sample collections with their chlorophyll data collections because I wanted to learn more about the carbonate chemistry in the area that I'm studying ocean acidification. And so what we found is that the pH varies with biomass. So on this y-axis, I have the pH on the total hydrogen scale. And you can see that right after this big phytoplankton bloom that happens as soon as the sea ice breaks out and blows away, you get a big surge in that phytoplankton population, and then you get a big change in the pH responding to that. So I use this information, or I'm highlighting this information because I used it to inform my experimental design. So for my temperature treatments, I set my ambient pH at 1.5 degrees C because, one, that was the temperature of the seawater while we were there for our field season. It also corresponds with around the median or mean temperature value for the summertime. And so we set our near future predicted temperature level at 3.5 degrees C, which is an increase of 2 degrees C, which is predicted by the Intergovernmental Panel on Climate Change, or the IPCC, using the business as usual models. So for our pH treatments, we also use the business as usual models, and we set our ambient pH at 8.0 because, again, that was the pH at the time that we were at the station, and it's pretty close to that median pH that these organisms experience throughout the year. And we set our near future level at 7.6, which is a decrease in 0.4 units, which is again based on the IPCC predictions. So using this setup, we have 4 different treatments, and I have labeled them as ambient, reduced pH, high temperature, or a combined treatment. And I'm showing you this notation so that you can see how those actual levels correspond to what I'm calling them. Everybody with me so far? Excellent. Okay. So I am interested in using multiple stressor studies like this where I can combine warming and acidification because you can get information about how these two potential stressors interact. So these interactions can either look like an additive effect where you have the net effect that you observe looks as if you would have just added the effect of A to effect B. So that's what you get when you have an additive effect. But that's not the only interaction you can have. You can also have a synergistic effect, where that net effect that you see is larger than if you just add effect A to effect B. So they're kind of multiplying each other and increasing whatever that effect is. But you can also imagine you can have the opposite, where one of them is inhibiting the other. So you can also have an antagonistic effect, where the net effect that you see is less than if you just added effect A to effect B. Okay, so that hopefully sets the stage. I, for my PhD, studied a whole lot of metrics with a lot of different organisms. Well, a lot is these 4 organisms were my target species. But today, because of time constraints, I'm going to focus on the results from my survival observations and where I measured biodiversity with these crustaceans, primarily these 2. So I'm going to introduce you to my players. So these are my lab animals. This one is Gondogenea antarctica, which in the fondly referred to as gondo. Gondo is an omnivorous amphipod. It's pretty small. They're very ubiquitous. You can find them just swimming around the benthos, but they primarily associate with these big browns. That's where they are the most abundant. In contrast, our other lab organism that I used is Paradexamine fissicata, which we call Paradex. Paradex is a much larger amphipod, and it primarily associates with this one red macroalga. This is Placamium cartilagineum. So Placamium is a very strongly chemically defended red alga that none of the other amphipods eat. None of the other amphipods want to hang out on it. They don't want anything to do with it. But Paradex does. Paradex actually consumes Placamium and sequesters the chemical defenses from the alga in its own tissues. So this is I'm trying to set the stage for a more generalist with a specialist. A small generalist that moves around, very energetic, and a more specialist amphipod that is more sedentary, tends to just hang out on this one alga. So this is the contrasting life histories that I was interested in for this experiment. So to get our amphipods, we go out in these zodiacs in all different conditions, and then we hop in the water and we collect So the way this looks, or this is what it looks like. So here we have Yusheng. He is scraping an alga off of the rock, very careful to not touch the alga or interfere with it too much, and let it drop right into that fine mesh bag. In that way, he does not disturb any of the amphipods or anything living on it, and that way we can capture them all and bring them back to the lab. And then we very carefully rinse all of the amphipods or anything on that alga off, and then we can go through and sort through and pick out our target amphipods. So that's what our collection looks like. When we bring them back to the lab, this is what our lab facility looks like. So for my PhD, this is the lab facility that we built at Palmer Station. This is a temporary setup that was only present for our experiments, and then we took it down between our field seasons. So we controlled temperature using these big water baths. We have 4 of the water baths that then control the temperature in these larger acrylic water tables. Each of these containers is controlled individually for its pH. So we had pH sensors, and we were able to bubble CO2 or air into each of these containers individually. When you look in each of these containers, this is what it looks like. You can see it's kind of crowded. We have it set up this way because I don't want my amphipods to interact with each other. I want to make sure that what I'm measuring is just because of how they're responding, not because of how they're responding to the interactions and my treatments. So in each of these little, what I like to call amphipod hotels, we had either 15 Gondogenea or 10 paradexamine, which should give you an idea of how their sizes differ. I had about the same biomass in both of those hotels so that you can tell they're different sizes. And then you also may notice that we have some macroalgae in here. That was a whole separate experiment where we're exposing the 2 big brown macroalgal species to these ocean warming and pH conditions, and I didn't want my amphipods to get really, really hungry and mess with the other experiments. So that's why we have them in these little hotels. And so maintain them in this experimental setup for 90 days. So now we're going to go into some results. So to start off with, I've got the proportion of amphipods surviving on the y-axis, and then I have time on the x-axis. And these are the results for Gondogenea. And you can see that the ambient and the temperature, high temperature treatments, those amphipods, there was not very much difference in their survival. However, as soon as you add low pH into the mix, my survival dropped dramatically. And there was a significant difference between the survival for the amphipods and the low pH treatments compared to the ambient and high pH treatment, or high temperature treatment. There were no differences between the temperature, or the ambient and high temperature treatment. You'll also notice that there is somewhat of a synergistic effect right here. So the difference between the temperature in the ambient, or the high temperature in the ambient treatment, is fairly small, but it's larger when you combine that low pH and high temperature. So these are the results for Gondogenea. When we compare those to paradexamine, you can see that paradex did way worse. They really did not like that low pH treatment. Even so, in fact, at the end of the experimental period in this combined low pH high temperature treatment, I only had 20% survival. I spent the entire experiment freaking out that there was something going wrong because I get attached to my organisms and I really wanted them to survive. So I was checking these every day. So I was checking their survival every day. I was also checking for molts. And one of the things that I noticed, these are the percentage of molts that I collected every day. So on the y-axis, I have the percentage of molts, and that's what each of these symbols represent, is the mean plus or minus the standard deviation. And you'll notice that there is an increase for both Paradex and Gondo around the same time period. This also corresponds with an increase in mortality. So what we found when we ran the stats, this was not a coincidence. This is happening at the same time. It looks like the amphipods were trying to molt, and then they weren't surviving their molts. So amphipods, their exoskeleton is calcium carbonate reinforced. And also when they're solidifying their exoskeleton, that's when their ion balance is the most in flux, and that's when they're the most sensitive. So I should have predicted this, but I didn't realize it was going to have this big of an effect on their mortality. All right, so I'm going to shift gears a little bit, and we're going to talk a little bit about, we're going to define some terms again. So people talk about resistance and resilience when they are talking about climate change a lot. And depending on the context, that can have different meanings, especially when you're talking about it in the context of ecology. That has a very specific meaning. So just to illustrate, I have made a couple theoretical diagrams to highlight some of the things that I want to discuss. So if you have some response, have a disturbance, there'll be some kind of, there can be some type of reorganization in response to that disturbance, and then the organism or community can rebound, and that's termed resilience, when you have this reorganization to get back to the original level. So if you have that same disturbance and there is no reorganization and there's no change in that response, then that's considered to be resistance. Okay? So resilience, there's a change, but it comes back, kind of like when you stretch a rubber band, you get some resilience, the response, and then resistance, you just don't change. Some of the things that can contribute to resistance are response asynchrony or synchrony. And so this is, again, another theoretical diagram that I made. So if you have survival, you have a community of organisms and several disturbances, and those communities respond differently, and then they, all of a sudden, that is considered response asynchrony. But if they all start responding the same way at the same time, then you have response synchrony. And if you look at a net level, this here looks like resistance, right? And then you have your potential resilience coming into play when you have that response synchrony, syncing all of those responses together. Does that make sense? Okay. All right, so I wanted to look at how resistant or resilient the crustacean assemblages that associate with the macroalgae that I study are to changes in pH. So at the same time that I was running the 90-day experiment with Gondo and Paradex, I was also running this mesocosm experiment. So for the mesocosm experiment, we collected the amphipods the same way that I did for my single species studies. We went out and we collected the macroalgae in those fine mesh nets, and then we rinsed off all of the amphipods. We didn't pick anything out this time, and we pooled all those amphipods together, and then we used a plankton splitter to make aliquots from that pooled population, and then we seeded those into each of these mesocosm buckets. So each of these mesocosm buckets were one of 3 pHs. It was either 8.0, 7.7, or 7.3. So that represents the ambient conditions. Our near future in this case is the 7.7 pH treatment, and then we pushed it to a very far future low pH at 7.3. So after 1 month, we ended the experiment and then collected all the amphipods that were still surviving, and then I went through and I identified all the species present. So this is what it looks like. So here on this NMDS plot, points that are closer together are more similar to each other, and those that are further apart are more dissimilar to each other. Each of these symbols represents one mesocosm, and it represents all the species that I identified in a given mesocosm. So I've got some lines on here to demonstrate the similarity between these mesocosms, and you can see for the most part they kind of group together. But when I ran an anosym or an analysis of similarity, I found that there was a significant difference based on pH. And so when we did the pairwise testing, we found that that was because the 7.3 pH treatment was differing from both the 7.7 and 8.0 pH treatments. So it looks like at near future conditions, these amphipod communities, dominated communities, are pretty resistant to the conditions that we presented with them. But then when you push it really far forward into the future at a lower pH, you get a reorganization of that community. So what do these communities look like? Here are the counts from my experiment. So on the y-axis, I have the mean number of that I counted, and on the x-axis, I have the actual species that I identified. And so I just want to highlight a couple things here because there's a lot. Each of these bars are color-coded based on the pH exposure. When you look at those species that are the most abundant in my treatments, you can see that they're not all responding the same way at each of those pHs. So I've got some response asynchrony among those organisms that are the most abundant. But that's not the only way you can parse these data. We can also look at it in functional groups. So when you look at the omnivores, all of the omnivores that I had in these treatments, they're not all responding the same way at each pH, and neither are the herbivores. So I've got response asynchrony at multiple levels, or different ways that you can look at these data. So overall, it looks like, at least at near future conditions, these amphipod communities are resistant to low pH. However, there are a couple drawbacks with this study, and those are predominantly dealing with the time period. We thought we were doing really well with having a 30-day exposure period. We thought we'd really catch a lot of the dynamics there. And then we only were looking at pH. When I mentioned 30 days, hopefully that raised something for you, because 30 days also happens to be right when we had the amphipods going through their molt cycle. So at least for 2 of the amphipods that we have in those communities, their natural history, they would be molting every 30 days. So I might be missing some of the responses. Maybe some of these amphipods in these communities never got a chance to get to a molt, and so they never had that same challenge that other species did. So it looks like 30 days is too short of a time exposure for these species. So what I was just doing for the last 2 months, we were down again at Palmer Station, and we were repeating, or we've modified the experiment a little, but we're looking at how these assemblages respond over a longer time period. So over 3 months, how will these assemblages respond? So my job for the last several months was to build this. This is a mesocosm setup where we have each mesocosm paired with its own mixing tank, so we can adjust the seawater pH for each mesocosm individually. And this experiment will run for the next couple of months. It was really satisfying to get this up and running, because I should have had before pictures. There's nothing here. Before we got there, it's just these big round tanks. So all of this infrastructure is what we built in the last 2 months. So it's been a really cool project, and I'm really excited to see the results. So what we're hoping to get from this is to identify a few species that are winners and losers. So we're going to the way we're going to assess that is through survival. So we'll find 2 species that don't do well at these pH treatment levels, and next year, and then we'll find 2 species that do well. And then next year, we're going to repeat these exposures that use individual exposures instead of the mesocosms, and try to get at the mechanism for why they either do well or they don't do well in these future conditions. So my work with climate change in the Antarctic has not only been in the lab, it's also had a field component. So again, I want to highlight some of the LTER data. So in this figure here, this shows the western Antarctic Peninsula. These colored dots represent stations. Every year, the LTER goes out in January, and they do an annual cruise, and they've been doing this for the last 30 years. They collect all kinds of data on everything from the bacteria up to the whales present in January. And they also look at sea ice cover. So that's what these little insets represent. So up here around Palmer Station, this is what the sea ice cover looks like. So on the y-axis, you have time from 1981 coming to the present in 2011. And then on the x-axis, again, is time, but it's in months. So it's going to show the seasonal changes. And the color coding shows sea ice cover. So the lighter colors represent more sea ice cover, and the darker blue colors represent more open water. So I'm showing you these because I want to highlight, up around Palmer Station, the sea ice cover has varied over time. However, if you kind of squint, it looks like those years where you have lots of sea ice cover, the frequency of those years is decreasing, and that sea ice cover is just not staying around as long. When you go further south, so the distance here is around 500 miles. If you go down to Rothera, which is the British Antarctic Survey's big marine station where they also do a lot of research, you can see this inset is the same setup as here, where you have time going from 1981 to 2011. Again, there's more sea ice cover because it's further south, but it varies over time. and it tends to be getting less. And we go further south to the southern end of Marguerite Bay, and again, more sea ice because you're going further south still. And but again, it's varying, and it's the frequency of the years where you have low sea ice is increasing. So I'm highlighting this, or I'm trying to emphasize this because I want to show that sea ice varies with time and space on the Antarctic Peninsula. And because we have the robust diving program through UAB at Palmer Station, we know what the benthic environment looks like around Palmer. We know that it's dominated by these big brown macroalgae that I talked about. We also know, because they have a robust diving program at Rothera, that this is what the benthos looks like. You have a lot less of those big brown macroalgae, and that's related to the sea ice. The sea ice not only shades all of the macroalgae so they can't do photosynthesis, you also get sea ice scour, where it scrapes everything up off of the bottom. So there's a lot less of these big, robust communities. However, we don't know what the benthos looks like when you go further south or anywhere in between Rothera and Palmer Station. We know, well, that's overstating a little. We know a little bit. So the last time somebody looked at the benthos between Palmer Station and Rothera was in 1973 when they took down this wooden vessel, the Hero, and they did surveys along the western Antarctic Peninsula. So I'm going to show you some of their data. I don't expect you to get a whole lot out of this. All I want you to see is the data that they were able to collect at that time was basically what species they could identify and then presence or absence at a given location. With how much sea ice dynamics are changing now, that's not enough. We need to know more about what these communities look like. So this last year, in May and April, we did a cruise where each of these green dots represents a dive site, and then the color gradation here represents sea ice cover. So we chose sites that go across the sea ice gradient, and we wanted to look at the benthic communities that are associated at these sites. And so we did all this on scuba. We did primarily our biodiversity counts are going to be based on video transects that we did. We did vertical and horizontal transects at each of these sites. We also did organismal collections where we collected the macroalgae and invertebrates present. We also did some suction sampling to get some of the smaller organisms that we might not be able to catch with the big macroalgae. We brought these all back to the lab where we dissected and collected material for genetics as well as biomarker analysis with stable isotopes and fatty acids. So the fatty acids is where my work comes in for this. So fatty acids can be useful as trophic biomarkers, and so I'm going to give you a quick reminder of what fatty acids are and their role as biomarkers. So there are fatty acids are the building blocks of lipids. Within a marine or aquatic organism, you can identify anywhere between 30 and 50 fatty acids. They can be grouped into several categories. You can have saturated fatty acids, which that's where you have no double bonds in the carbon chain. You can also have monounsaturated fatty acids, or MUFAs, which is just fun to say. You have one saturated, or you have one double bond in that carbon chain. Or you can have polyunsaturated fatty acids where you have more than one or more than two double bonds in the carbon chain, and those are called PUFAs. So this is one example is EPA. You might have heard about this. It's often advertised in fish oil supplements. So fatty acids can be, or particularly omega-3 fatty acids and omega-6, or particularly omega-3 fatty acids, they can be synthesized by primary producers. And so knowing this, we can trace the fatty acids from primary producers up through different organisms by looking at the suite of the fatty acids that you can identify. You can use all of those fatty acids as a signature. I like to think of it as like the points on a fingerprint when people are when the police are trying to trace down somebody. But you need to have a little bit of information about the fatty acids and how they get trophically modified, because as you can imagine, when you eat something, your body does not turn into like a piece. If you eat a piece of lettuce, your arm's not going to look like a piece of lettuce. You're going to have your own physiological needs that need to be met, and so that will mean some type of breakdown or synthesis of that material, and that includes the fatty acids. And so with feeding assays, you can look at the fatty acids of the food that they consume and then what fatty acids are retained. So I'm highlighting all this because this is one of the core parts of a project that I had funded recently, where we've been looking at the responses of juvenile Dungeness crabs to ocean acidification. You guys all aware of Dungeness crabs? They're pretty great. All right, so Dungeness crabs, they're really abundant on the West Coast. You can find them from Alaska all the way down to California. They live in seagrass beds all the way down to around 300 feet. Usually the juveniles recruit to the seagrass beds, and then they live their first couple years there, and then they migrate out. One of the reasons why it's interesting to look at Dungeness crabs is when they're recruiting in, to the coast, that's when you get a lot of the seasonal upwelling. So they're exposed to this naturally low pH water. Dungeness crabs are the biggest fishery in Oregon by amount of money that it brings in and biomass. So this is a really huge, important deal. But people haven't really studied the effects of ocean acidification on this species. You probably are familiar with crab life history cycles, but on the West Coast, Dungeness crabs, they reproduce. The females brood the eggs. Once the eggs are released, they get carried out to the open ocean, where they go through several larval cycles before they metamorphose into megalopy. The megalopy are then recruited back into the coast, where they will settle as juveniles, and then they'll live along the shallow benthos for 1 to 2 years, and then they'll migrate deeper. Often when they're in the juvenile form, they hang out in estuaries. So I am really interested in how ocean acidification influences molting, and molting for crabs is also a very stressful time period. And so during the larval stage and juvenile stage, this is when they're molting the most. So a lot of models that have predicted that Dungeness crabs will be resistant to ocean acidification have all been focused on the adults, and that's because the adults are not molting as often, they're not growing as often. And so that's why we've chosen to focus on this juvenile stage. And so when we get a recruitment, this is what it looks like. We have a colleague who has a light trap he puts out every year and collects these megalopy. So on this video, hope you'll be able to see it well. All these little guys right here, these are all juvenile Dungeness crabs running away from our inexperienced ROV operator. This is slowed down 50% so that you don't get seasick. But there are tons of these crabs, and they're running away from this video. And this is just in the boat basin right by our marine lab. So one of the things that they occur in high densities, but they rely on their sensory systems to stay away from predators, even if those predators are an adult crab. So you can see that there's a little halo that these little crabs keep around this Dungeness adult, so that they're really relying on these guys, their sensory systems. So what we did is we collect the megalopy, settle them as juveniles, and put them into an experimental facility that I've built at the Oregon Institute of Marine Biology. So we're able to control the pH by bubbling CO2. We filter our water. This is the little, what I like to call the brain box, where we control everything. And this is my intern, who I've been working with for the last year. She's been instrumental in helping me build all this. But this is what it looks like in one of our replicates. We have these little juvenile crabs that we're raising, and then we'll look at a bunch of metrics after we expose them to our experimental conditions. So those metrics include the fatty acid biomarkers that I mentioned, as well as growth and molt rates, and some behavioral metrics. And so this is ongoing work. Those molting is really important for helping us figure out if they're delaying their molts, and so therefore delaying their growth because of the added stress with the low pH. So that's one of the reasons why I want to highlight that we're looking at molting. We also are looking at behavior. These are 2 behavioral flumes that I had my REU students last summer built. Here is just food coloring dropped in the water to demonstrate that they achieved laminar flow through these flumes. And basically, the question that one of my students wanted to see was whether crabs would go away from a low pH condition. Do they actually not like that, or do they care? Because they are living in estuaries where they are exposed to low pH. And we found that they do run away from, they prefer to not be at 7.4. We also did, my other student did some studies with food searching behavior with the juvenile crabs in the flume that she built. So we're working on fleshing out some of those projects. So in the future, hopefully I've made the case that with the tool set that I have presented, that moving forward, I would like to continue doing similar research, and that I'd like to look at the responses of individual species and to ocean acidification or multiple stressors such as warming or hypoxia or freshening. I'm also really interested in the chemical ecology, how chemical sensing will be affected when you change the abiotic environment with warming or pH. And then I would like to continue working with groups of organisms and figuring out how their trophic interactions may shift in a changing ocean. So with that, I want to say thank you to a lot of my funding sources and the people who have supported this work. So for my Antarctic work, UAB and SIGMA-SCI as well as NSF supported financially a lot of that work. For the cruise that we did last year was NSF funded and supported, was a collaborative effort across several institutions. And then the Dungeness crab work I've had funded primarily through Sea Grant, but I've gotten additional funding from the Murdoch Charitable Trust as well as the University of Oregon. So all of this work has been done by a lot of amazing, I've had the help of a lot of amazing people. These are some of them, and any acknowledgement slide would not be complete without thanking them explicitly. And so with that, I would like to take any questions.